Hey everyone, we're going to look at part 4 of our cells unit. We are going to focus today on the mitosis detail, and I've got a plant cell drawn this morning, and I know that it's a plant cell because it has a cell wall around the outside, right? In terms of just review, where are we now? So our first video, oops, sorry, we're sliding around a lot there. In that first video, we looked at types of division, right? So we looked at this would be mitosis, and I know it's mitosis because there's a nucleus right there, right? So this is a multicellular, a larger organism. My pen is not working very well. So this is a mitotic division. I have a nucleus here, and a cell goes through a division process to get two identical daughter cells. This is asexual reproduction. We also have budding, which looks something like this. So you have a yeast cell, let's say, and it kind of pinches off this little tiny bud. There's a lot more that goes into division than that. So if you don't remember those things, go back and take a look at that first video in this set. Then we've got the cell cycle. This was part 2 of our series. Cell cycle, right? And the cell cycle, we've got 3 phases essentially. So we've got G1 or 4 really. Synthesis phase where DNA is replicated, and then G2. And then mitosis happens right here, and then we've got cytokinesis right there. So our cell is actually splitting into 2. The cytoplasm is dividing, and the organelles are assorting themselves. And then in the third set, we focused a little bit more in on the cell cycle, and we looked at regulation of that cycle. So the regulation comes in many different forms, but this would be one of those checkpoints that we talked about. So a cell reaches this spot, and if that checkpoint is good, it will carry on through. If it is no good, if that checkpoint is not met, or if the conditions for that checkpoint are not met, then the cell will either try to fix the problem, so maybe it grows some more, or it checks this DNA replication before it goes through that division. Also remember that there is significant regulation in the S phase, in the cell reproduction, in the DNA replication stages. That's chapter 12 in the OpenStax textbook if you need to go back for a refresher. So that's where we are in terms of cell cycle, right? So today we're going to focus a little bit more specifically on mitosis. The other term for this is karyokinesis, and karyokinesis is simply the splitting of the nucleus, right? So karyo, that's a little bit hard to see, so let's get rid of that. Karyo means seed, it's Greek. Kinesis means to move, so when they were looking at mitosis, they saw this little thing called the nucleus, and it looks kind of like a seed, and it split, it moved. So karyokinesis, same thing as mitosis. But the main idea is that we have a division with 2 identical daughter cells being formed. And here's the key, right? It's asexual reproduction. We are forming brand new cells. And we split mitosis into 5 different phases. So let's take a look at those. So the first one up is prophase. And the indicators for this are the nuclear envelope, or the nuclear membrane, disappears, right? It goes away. We need to get those chromosomes out so we can segregate them. And the spindle fibers appear. And spindle fibers are grown in an animal cell by these things. This is called a centromere, or a centriole, I'm sorry. And a centriole produces these spindle fibers. You can see them starting to form in this picture. And a spindle fiber is some protein, and it's like a microtubule. So it's propagated by the centriole, and that is what does the pulling. It's the separation mechanism. Now there are no centrioles in plant cells. These are in animal cells only. And the interesting thing is, we're not really sure where spindle fibers come from in plant cells. There's some research that shows that the centromere, the point of attachment, so if we're looking at our chromosomes, right? So we've got chromosome A, chromosome B. Okay, the centromere is that attachment point in between them. There is some research that says that the centromere is what actually propagates the spindle fibers in plant cell mitosis, so this would be spreading out. But there's not a lot of evidence about where those microtubules come from. So there is some kind of different mechanism in plant cells. Spindle fibers are the same worker in that mechanism, but there are no centrioles in plant cells. So be very, very careful when you are when you're describing mitosis. So that's prophase, so the nuclear envelope disappears, spindle fibers appear. Prometaphase is kind of this in between. So we've got the spindle fibers attached to the centromere, right? So they're attached to that central region on the cell. The chromosomes are fully condensed. but they're not aligned yet. So if you're looking at your cell, you should be able to visually recognize what is what. So this is kind of a point between prophase and metaphase, hence prometaphase. And so we're moving toward the metaphase stage, where our chromosomes align on the cell midline. So we see individual chromosomes. So I should add this, because in meiosis it's a little bit different. So these are individual. Okay, and remember our chromosomes are doubled. Okay, so we've got 2 drawn here, but we have individual chromosomes aligning on the midline. Spindle fibers are still attached to the centriole because I've got an animal cell drawn. In anaphase, spindle fibers are drawing the sister chromatids to the poles. So we've got the chromosomes here. Remember, they're paired up, so these are called sister chromatids. They're homologous. That means they have the same genes, and they're drawn to the poles. They are segregating. And I should say that these stages, metaphase, anaphase, and telophase, are the fastest of the cell of mitosis, because if I'm moving my chromosomes around, this is all of my genetic material locked into that chromosome, right? So I don't want there to be any chance for that chromosome to become damaged. So when spindle fibers draw the sister chromatids to the pole, it happens very quickly. And we draw them like this because if you imagine pulling a piece of paper or a string through water, right, this is full of cytoplasm. And so those chromosomes are kind of dragged through, pulled back to either side of the cell. And as soon as they're there, the nuclear envelope begins to rebuild itself. So in telophase, the spindle fibers, they disappear. Okay, the spindle fibers are reworked back into the cytoskeleton. So this is the internal structure of the cell. Protein-based, so that cytoskeleton begins to reform. The nuclear envelope begins to reform because that protects our chromosomes. And so as that begins to reform, we start to form, this is called a cleavage furrow. That cleavage furrow, I'm writing that, is essentially, F-U-R-R-O-W, this is essentially like a zipper. So that nucleus, or the cell begins to form this cleavage furrow, and that will slowly pinch off the cell until we get daughter cell A, and daughter cell B goes on their merry way. They have exact copies of the chromosomes. Remember, genetic variation in asexual reproduction, like mitosis, comes from mutations, so there might be some variance in terms of base pair sequencing, but for the most part, these are identical cells. And they go on their merry ways, and then they continue to grow and do whatever. Also, don't forget that some cells, they enter what is called G0, right? So G0 is when we are not actively preparing to divide, nor are we dividing. So that'd be like a red blood cell, a cardiac cell, a neuron, things like that. Once they divide, they're done. So that's mitosis in detail. You should be able to describe what happens at each phase. So at prophase, what's going on at prometaphase, metaphase, anaphase, telophase, what's happening in each of those? What are the major indicators for each? Thanks for watching, and I'll see you next time.